In the previous video, we kind of worked through how all the ducts that the sperm is traveling through to go out, and there really wasn't a nice picture, so I just wanted to make sure that you were aware. In your book, page 1039, is a picture showing all of those ducts and kind of how they just lead into each other. But those pictures also have a lot of other anatomical structures listed, so don't get too lost. But I think it is worth a look to kind of look how it travels through the testes or out of the testes, the epididymis, and the ducts, and all the way out the urethra. So I would take a look at that. Page 1039. This video is going to focus on puberty and maybe the beginning of producing sperm or meiosis. So when we look at puberty, it's going to be a little bit different in boys and girls. So we're going to focus on boys here. But the reproductive system basically is dormant after birth, and so 10 to 12 years in boys. And then the surge of pituitary gonadotropins is what kind of awakens the reproductive system. And when that happens, it's the onset of puberty. And so that's the first few years of adolescence, or until the first ejaculation of viable sperm in boys, usually about age 13. So there is actually testosterone secreted in the testes in the first trimester of fetal development, but then dormant until puberty. And so then from puberty through adulthood, the reproductive function is regulated by hormones from the hypothalamus, the pituitary, and the gonads, which we touched on a little bit last chapter in the endocrine system, but we're going to focus on it again here. It's just the ones that have to do with the male reproductive system. So the hypothalamus, as it matures, produces this gonadotropin releasing hormone, and then that stimulates the anterior pituitary to secrete FSH and LH. And so in males, the FSH basically stimulates those cells to secrete the protein that binds to the testosterone, and that holds it into the seminiferous tubule, and that stimulates the process of making sperm. And the LH, the luteinizing hormone, stimulates the cells to produce testosterone. So both of those working together. During puberty, we would expect to see enlargement of secondary sexual organs. We would expect to also see testosterone to stimulate some other body growth, not just in the reproductive system, but limbs, muscles, and even the larynx. And then we would expect to see pubic hair, some scent and sebaceous glands develop, and then stimulate sperm production and libido. In adulthood, we're going to sustain the sperm production and libido with continual testosterone. But the regulation of sperm production is kind of separate. That doesn't affect the hormone balance. So inhibin is a hormone that can suppress the FSH without reducing the LH. So basically we can stop the sperm production without reducing the testosterone secretion. So again, I don't want this picture to kind of freak you out, but see how that inhibin in the middle is kind of separate, and so they are not all working together. I mean, they're working together, but we can stop the production of sperm separately from reducing testosterone levels. Okay, so the production of sperm is called spermatogenesis. It is through the process of meiosis. So I'm assuming back in AMP1 that you covered mitosis and meiosis. Since I have Dr. Lynn's stuff from AMP1, I'm assuming that's what happened. But we're going to go through meiosis one more time here because it's going to make more sense, and because it's exactly how sperm and eggs are produced. So we're going to, I'm not going to really compare it to mitosis, but we are going to go through meiosis here. So it's important to know the three kind of main parts or events. Those large germ cells go through meiosis, and they, at the end of meiosis, they are small, mobile sperm cells that have flagella. They're able to move. The other big thing that happens during meiosis is the reduction of the chromosome number. That is super important. All of our sex cells only have half of the chromosomes of a normal cell. So a sperm cell only has 23 chromosomes. And then the shuffling of the genes that happens during meiosis. Sometimes we call this genetic recombination. It just makes sure that all of the sperm have different combinations that are different from the parents. This is very important for offspring to be genetically different from their parents. And so form 4 sperm cells come from 1 germ cells in meiosis. Meiosis always produces 4 cells. So this is just kind of a generic meiosis picture. So we start with paired chromosomes. The chromosomes duplicate before meiosis starts, and they are in pairs. But by the end of 2 cell divisions, we have 4 cells that are called haploid. That means they have half of the genetic information or half of the chromosomes that the original cell had. And so even though those chromosomes look different, I can count 4 chromosomes in that first picture in prophase 1, and then I only count 2 in each of the 4 cells. Now I know that some look like X's and some look like little gummy worms, but there is a reduction of chromosome number by half during meiosis. Again, I'm not going to go through mitosis, but every other body cell goes through mitosis. The only cells that go through meiosis are in the testes and the ovaries, and they are producing gametes. So they are producing 4 gametes that each have only half of the DNA of a normal body cell. When those 2 gametes meet each other, when the male gamete and the female gamete fertilize each other, then it goes back to that full number or the same number of chromosomes from the parents. So a sperm with 23 chromosomes meets an egg with 23 chromosomes, and then we're back to our magic number of 46. You should definitely watch these animations here and watch through the phases of meiosis. But really the most important phase of meiosis is prophase 1. And that's because in prophase 1, the pair of homologous chromosomes, they line up right next to each other and they do a process called crossing over. Crossing over creates that new recombination of genes, and it just basically ensures that all of the sex cells that are created are different from the original parent cell. This is how we have all of these different gene combinations that we see among our very diverse species. So you definitely should watch this. This just shows you an up-close visual of crossing over. Remember, each little black section is a gene, and that gene really can, we think about genes having one trait or making one protein, but really a gene can be in charge of hundreds of things within the body. So as those genes kind of swap places and cross over, it is a big recombination of genes. I'm going to let you watch through all of those. Again, I wouldn't be so picky about mitosis right now, but you definitely want to focus on meiosis. So then this is what meiosis specifically looks like in the testes. And so this shows the type of cells that would be going through meiosis. And it starts at the bottom of the picture with number 1. And so that type A sperm is going to stay there. Actually, let me go to the description here because I find this picture to be a little bit confusing between type A and type B. It makes it look very like they're identical, but they're not. So type B is the part that's going to go through meiosis, and then you can see as meiosis occurs, what we end up with at number 4 and number 5 are sperm cells that are ready to leave. Okay, so when we look at where these sperm cells are going to, or where these germ cells, I should say, are going to go through meiosis, it is in the periphery of the seminiferous tubules. They're going to divide there by mitosis, and in mitosis, we're going to have 2 daughter cells. So one type A cell is going to stay in the wall, and then the type B cell is going to move away from the wall, and that type of cell is going to go through meiosis. So I didn't really like this picture, how it looks like, let me go back, how it looks like they're both the same. It kind of looks like the type A turns into the type B by the fact that that arrow number 1, that's not exactly accurate. So when they divide by through mitosis, they are identical, and so there's 2 cells formed there. One's going to be a type A and one's going to be type B. Type B is the one that's going to move away from the wall, and it is going to go through meiosis. So that becomes a primary spermatocyte, and then it's going to go through meiosis 1, and that's the first cell division, and then it's going to have 2 secondary spermatocytes, and then they're going to go through meiosis 2, and at that point they're going to each make 2 spermatids. Okay, so that's 4 total, and they are not going to divide any further, but they are going to kind of transform and differentiate, and they're going to develop into sperm. And this is basically showing that same exact picture, and shows where in this seminiferous tubule are the sperm finally mature, and that's all the way towards the inside. Kind of deciding if I need to wrap this video up or if we can make it through. Let me just kind of peek how much more. All right, I'm gonna end this here and I will pick up in the next video.